Hi everyone, and welcome to our video show on enzymes. You can tell by my first slide here that enzymes are, what are they? They're protein. So let me just write that here. And from what you know about proteins, meaning their monomer is what? Their monomer is an amino acid. And the most important thing about a protein is its shape. And same goes for enzymes. Since enzymes are a protein, their shape is super crucial to all their functions. Now, enzymes are crazy because life literally could not exist without them. You might have heard enzymes called biological catalysts. Biological catalysts. Put little quotes around that. So you might remember this word catalyst from chemistry class. It's a substance or a chemical compound that breaks down or puts together or speeds up a reaction without itself being consumed. Well, that's the same thing with enzymes, but enzymes are found in living things, and they're made of different things than the traditional catalysts that you've learned about in chemistry. So they're made of proteins, right? Enzymes are incredibly crucial to life and to the function of living things, and we're going to dive into that right now. So here is a picture of a human, and there is an enzyme, well, found in pretty much all living things, found in fungi, plants, animals, bacteria, and it's an enzyme called catalase. Let me spell that correctly for you here. Catalase. Now, before we move forward, I want to tell you a little hint. Now, we know that when we learned about carbohydrates that the monomers always ended in ose, O-S-E. Well, enzymes always end in A-S-E. So we have catalase, lipase, amylase, protease. They all end in ASE and have that A's kind of sound. So this specific enzyme, catalase, is found pretty much in all living things, and it's highly concentrated in the human liver. Okay? So the function of catalase is it has an active site, which I'll talk about later, that takes this molecule, you guys know what this is? This is hydrogen peroxide, and breaks it down into H2O and O2. Okay? So many of you know that hydrogen peroxide is not necessarily something you want to have in your body. Okay? This is considered toxic. It's a toxin to our body. I know if your dog eats something bad like chocolate or gets into something it shouldn't, you're supposed to feed it 2 tablespoons of hydrogen peroxide and it makes him throw up. Not sure if that's a tested thing, but something I read in a book somewhere. Let's go ahead and balance this equation first. So we have 2H2O2 and 2H2O. There we go. Got to be balanced. So this is toxic, and this is actually produced, hydrogen peroxide is actually produced in the liver as a byproduct of breaking down other toxins. Now you know that liver, its primary function is to break down toxins in the body, especially alcohol, and a big byproduct of this is hydrogen peroxide. Now, we don't want to have high concentrations of hydrogen peroxide in our liver or in our body at all. So we are super stoked that we have catalase, our friend, that actually gets this reaction moving, and it moves really quick. So we have our hydrogen peroxide ends up becoming much less harmful things. We get 2 molecules of water and 1 molecule of oxygen gas out of this toxic thing. It's also thought that recent studies, people think that individuals that lack this catalase, it can actually cause gray hair. So the idea that the hydrogen peroxide, the higher concentrations within the body and within the bloodstream actually bleach the hair from the inside out. So that's very recent studies. I'm looking forward to seeing more information about that. So we know that they're important. So let's look at exactly how they work. So you might have seen this before. This is a graph of a reaction. So here we have energy. This is my freehand for energy right here, my shorthand. And here we have the progress of the reaction. So going from, let's see, we are going from reactants all the way down to when we're done with the reaction, we're at products. Okay, and our red little line shows us the reaction. I'm going to abbreviate reaction RXN. This is the reaction without an enzyme. And this black line right here, this is the reaction with an enzyme. Hope you're understanding my shorthand here. Okay, so if this represents energy, and we see that no matter what, energy has to be an input for a reaction to go from reactants to projects. And I call this energy hill. Energy. So no matter what, a reaction has to go over this hill. Now, what's cool about it is an enzyme is it makes this hill, this amount of energy required to get a reaction started, makes it much smaller. So enzymes actually lower the activation energy. Energy. So they make this energy required for a reaction to go from reactants to product. It takes it and it reduces it. So if this right here, from this line to this line, this represents the energy needed without an enzyme. And this little distance from here to here, which measures from the top of this hill to the bottom of this hill, is energy needed with enzyme. So enzymes basically allow reactions to happen much easier. Just like if you were a hiker, right, it would be much easier to go up this little guy right here, which might be Bishop's Peak or Islay Hill in San Luis, rather than climb this guy, which might be something like Mount Whitney, okay? So enzymes are super important because reactions can happen a lot quicker and more often with them because we are requiring lower activation energy. I'm going to put a little box around this. Okay, so let's talk about the physicality of an enzyme. So this is a very, very simple drawing here of an enzyme. So this would represent the enzyme complex. Now, if you think back to proteins, you'd know that this would represent a quaternary structure of a protein, which may include 2 or more tertiary structures made from individual amino acid chains. So if this is our enzyme, which is a protein complex, this area right here I'm going to outline it right there. This is what's called the active site. This is the site on an enzyme that is going to be where the substrate attaches and where the enzyme performs work. So let's look at a little more complex image of this. Here we go. So we've got a few steps here. So this right here is our enzyme. And this right here is our substrate. Now, the substrate is just the molecule or the item that an enzyme is breaking apart or putting together. So it's whatever the enzyme is acting upon. Okay, so here we have our enzyme, here we have our substrate, and here we have our active site. Okay, so we could kind of look at this like the hydrogen peroxide. So if this is catalase, which is found in high concentrations in our liver, and this is hydrogen peroxide, the hydrogen peroxide molecule would have a specific like physical geographic location on that enzyme, that protein, that it would attach to. Okay, and we call this the induced fit model. Now, what's meant by induced fit is the enzyme slightly changes shape to accommodate this substrate. So I'm going to write that down here. So enzyme changes active site. My dots on my eyes are quite pathetic. Slightly. So if you notice that there's kind of this like blah shape right here, and as the substrate becomes attached to the enzyme changes, the enzyme active site changes to fit that substrate better. That's why it's called an induced fit model. The very presence of the substrate makes the active site more accommodating, changes the shape of the active site so that the enzyme can fit better. Okay, so then the enzyme acts upon the substrate, and here in this step we have the bond is broken. And on this step we have our products are made. Yay! Okay, so if we look at this like catalase, so this is catalase, we have hydrogen peroxide here, and at the end we would have water and hydrogen gas. Okay, now we use the induced fit model as the most widely accepted model. You might have heard of the lock and key model. Now that model illustrates the idea that the substrate and the enzyme fit very specifically like a lock and key, and that the active site does not change, that it's always at exact size and shape and fit. But the induced fit model is more widely accepted in scientific communities. So, we know enzymes are proteins, and because they are proteins, we know they are subject to become denatured. Okay, so a denatured protein does not work. So denatured enzyme does not work. I'm going to talk about 4 things that alter the effectiveness of an enzyme. These 2 can lead to denaturation. So each enzyme has its optimum conditions that it can operate in, right? So an enzyme that is within a bacteria that lives in a vent under the seafloor where it can get really, really, really hot has an optimum operating functional temperature a lot higher than enzymes that are in our bodies, right? So 98.6 or so degrees. So a big thing that can affect the effectiveness of an enzyme is temperature. Now, if an enzyme gets too hot, the protein unravels and its shape is no longer functional, so it can no longer put apart or together substrate. Another thing that can mess up an enzyme is pH level. So if it becomes exposed to acidic conditions or to basic conditions. In our body, enzymes really like to be around this 7 range. They love it there. But in enzymes that might function in animals that might be halophiles, they love salt, or live in crazy pH conditions, would be able to operate on a much higher or much lower range. So those are two things that could denature an enzyme. Now, I apologize for the poor quality of this image, but it's actually quite a really good image, so I wanted to include it anyway. Another thing that affects the effectiveness of an enzyme is the substrate concentration. Now, you remember that the substrate is what the enzyme is breaking apart or putting together. So here, if you follow along this line, we see that when we have low substrate concentration, so if these little guys, these little circles are our substrate, when we don't have a lot of substrate, we don't really have a fast reaction rate, okay? So reaction rate is on this side. So when we are lacking substrate, our enzymes are just kind of hanging out, waiting around for substrate to come around so it can break it apart or put it together. As our substrate increases, our reaction rate increases. The cool thing about enzymes is they're not used up. So a substrate can come, park it in the active site, and then leave as its products, right? And then the enzyme is just, all right, next, come on, come going. And this can happen incredibly fast, so ridiculously fast. So it's really awesome that these things are created and they're coded in our DNA so that they can speed up these reactions, like breaking down hydrogen peroxide in our liver or digesting our food. So substrate concentration is a way that we can change the reaction rate of our enzymes. And another thing that, well, if you think, okay, substrate concentration affects the reaction rate, well, enzyme concentration affects the reaction rate. And this is kind of a bit of a no-brainer. The more enzymes we have, the faster the rate of the reaction. Even though they individually work really quick, the more that we have collectively working together to break apart or put together substrate, the faster the reaction is going to go. So what we have here is a phenomenon in enzymes called competitive inhibition. Now, competitive inhibition is caused by this kind of evil little molecule called an inhibitor. And I'm going to put, there we go. He's got to dress the part, right? So the inhibitor molecule basically clogs up this active site right here. So the active site is where the substrate would normally connect to the enzyme. So the inhibitor kind of hangs out in this active site, not allowing the substrate to interact and to either be broken apart or put together. Okay, so another model is the non-competitive inhibition. So non-competitive inhibition. There we go. Lots of I's there. So in a non-competitive inhibition, here's our enzyme, here's our substrate. The substrate bonds to the active site, and now we have the substrate broken into its two molecules, Pac-Man and a diamond, right? In non-competitive inhibition, the inhibitor, this is our inhibitor molecule right here, it doesn't hang out in the active site. What it does is it goes to another part of an enzyme, and here's a picture of it right here. So it binds to a part of an enzyme other than this active site, okay? Now, the bonding of that actually changes the physical shape of the enzyme so that the active site no longer fits the substrate, and the substrate's really confused because it cannot fit in there. Therefore, it cannot be broken apart, okay? So the way I remember this is non-competitive. The inhibitor is not competing for the active site. In competitive inhibition, the inhibitor is competing for the active site. Now, let's take a look at some important enzymes in our body. Here's a mite. And we have an enzyme called protease. There's that A-S-E again. Now, if you can dissect this word, you see that protein kind of sounds like protein, and protease is a digestive enzyme that breaks these bonds. What are these bonds called? These are called peptide bonds. And what are these called? These are our amino acids, right? Our building blocks of proteins. So protease is responsible for when we eat food, and when we eat protein, protease is what breaks those peptide bonds so that we have our amino acids, our little building blocks to build new proteins. Another enzyme that is pretty important is called lipase. And if you can kind of dissect this word, this breaks apart bonds in fat molecules. So lip meaning lipid. So that is breaks apart fat molecules. And if you remember, those are ester bonds, bonding the glycerol to the fatty acid. And one that you're actually producing right now, well, you're producing it if you're eating, is a enzyme called amylase. Now, when you have food in your mouth and you're salivating, you actually salivate more when you eat. There's an enzyme in it called amylase, and it breaks down starch. So we're just going to imagine that this is thousands and thousands and thousands of glucose molecules long, and this amylase breaks down these glycosidic bonds between the glucose molecules and gives you some disaccharides and some really, really short polysaccharides. Okay, so there we go. There we have our enzymes and what they do and why they're important and why we cannot live without them. Happy biology studying!